Thanks for coming. I guess the selection committee made a mistake, but I'm not giving the award back. Sorry. So I'm keeping it. Good news is I have 300 slides. Better news is I'm not going to use them. Okay. I actually don't know what I want to talk about, to be honest, right? And with a few students, we were talking at lunch. Let's say I have never ever prepared for a talk except for my thesis defense here at U of M. But if I prepare for it, it comes out very artificial, and I always screw it up. So you just do it on the fly. It's going to be like that today as well. I want to tell you a little bit about what we are currently doing for cancer screening, and very much like to turn this into an interactive thing. So I want you to criticize it, ask questions, especially difficult questions, things of that nature to make it more interactive. And essentially what ends up happening is maybe I'll talk for 10 minutes, 15 minutes, and spend 45 minutes on question and answer, and that becomes the talk, and we'll answer the things that are important to you. Regarding this work on cancer screening, and what does I am a hardcore electrical engineer, circuit designer, chip designer, signal processing person. What does that have to do with cancer screening? Why? But the whole thing started with a actual tragedy in my life. It was a loss of my young and beautiful sister to cancer, and that drove me to develop this passion for trying to figure out what can technology do for it. And so I ended up investing in cancer companies, ended up interfacing with some great scientists, great biologists and biochemists, and learned a tiny bit of biology from them. And I started thinking as this engineer who was trying to learn biology and do something about cancer. Of course, this was a passion for years when I was at Broadcom. And it sort of continued until 2016, where this passion got so intense that I thought, oh, I owe it to myself to quit my job, despite discouragements from friends and family and everyone else. They thought I was crazy. I was the chief scientist of central engineering at Broadcom. Broadcom is 8 times, 7 times bigger than Intel now, and then almost $1 trillion company. I had the dream job there, and I quit to do this. This was all because of the passion and because I thought I really owed it to myself to try this. Otherwise, I would be sorry not doing it. And I couldn't be any happier today that I did that. And it is quite interesting that such a tragic event in your life can end up in something that makes me happy every day. I don't know how to wrap my head around that, actually. Never will. The whole idea is that I don't think anyone will argue with you that one of the best ways to fight cancer is early detection, right? I have witnessed that with my other sister, who also had breast cancer twice, but because of early detection, she's a survivor, no signs of it. My sister who died and my father who died, they didn't have that opportunity. Their cancer was detected too late. So this has been a personal experience. I don't need data to convince myself. I had real evidence myself early in this work. But I've never seen anyone argue with me that, no, early detection of cancer is a bad thing. In 3 cancers, breast, prostate, and colon, if you're detected stage 1, you're essentially saved. Other cancers have different degrees of being able to recover, but regardless of the type of cancer, detecting it early is always helpful. It's helpful in treatment, it's helpful in giving you a better chance to live longer, all of that. So there's no argument about that. But in order to do that, right, cancer, for all practical purposes, as far as we are concerned, is a random event. Even internal to your system, if there is something systematic going on, but this is still a random event when you detect it. You don't know when you're going to find that out, right? A random event, you got this edge and you want to sample it. The best way is to sample it as frequently as possible to be able to catch that, right? So if I do screening every 5 years and 10 years in between, God knows what's going to happen. In order to do so effective screening, you have to do this periodically. So where was I? Screening. For a screening to really be effective, it has to be periodic, right? The goal that was set from the very first day the SpyTech company was created was a $50 cancer screening cartridge, a home test that you can do every month from a finger stick, right? To monitor yourself for the onset of cancer as early as possible, right? Nearly impossible goal, and for that reason, for the first 5 years of the company, I did not look for investors because I didn't want them to have doubts or laugh at me or whatever. I financed the company myself until then later on when we started talking to investors. So that's a very tall claim, to do all of this stuff, get it done for $50, a cartridge that can work much like the COVID test that we are all familiar with. COVID, despite of all of the disasters, difficulties, and everything else that it brought to us, had this thing that everyone is now familiar with, this concept of receiving cartridge and testing at home. And this is not a foreign concept to them. It's quite acceptable for people to do that. And we want to get this cartridge for less than $50 to the end user, right? And that's where the challenge comes from. If you had enough money to pay $10,000 for a bunch of tests and whatnot, you know what to do today to go see if you might have cancer or not. But who's going to do that routinely? What insurance company is going to pay for that routinely? And this whole idea of making it cheap and affordable is a main element of this. Of course, on top of that, it must be reliable. It must be easy to use, right? And also, you have to have a plan. So what happens when it's positive? So what do you do now, right? So these are all the factors that we have thought about for years and years on how to put all of this together. This is, so I told you I won't use the slides, but I have lied before too. So maybe one or two. So this, where did it go? This title slide that you see there, this was 8 years ago. So this hasn't changed. This was the goal from the very, very beginning with that focus and that vision, which hasn't happened yet. But of course, at that time, this box didn't exist, that cartridge didn't exist, that title was there. And we sort of have paved the way for getting there, but I'll be the first one to tell you that it's not there yet. We've been operating in a stealth mode for all of these years, and not too many people know about us. We don't even have a website, we don't advertise, we don't hype it. This is a very sensitive area, thanks to that company, Theranos and Elizabeth Holmes, that screwed it up for the rest of the field. People have doubts, and they should, right? And that is why, especially coming from the engineering background, I gotta have the final product in my hand. When I tell you I have a cancer screening thing, so here, you go test yourself, right? And we're waiting for that too. We have enough patience to do that. And then you also have to go through all of the regulatory things that you have to do and deal with FDA trials and everything else to eventually have a useful product that people will trust, obviously. So we are on that path. But in order to do this, maybe I'll do this full screen. These are not a bunch of just beautiful pictures that you put together on a slide. Right? Every single one of these fields, from chemistry, biology, electronics, software, regulatory concerns, DSP and AI, electrochemistry, sensors, mechanical design, microfluidics, system engineering, and connectivity, they really have to come together as a totally multidisciplinary effort to make this possible. Just one innocent-looking cartridge that you saw on the first slide. Obviously, extremely tall claim, extremely challenging, which makes it fun at the same time. And you got to have a lot of patience dealing with this, because there will be disappointments along the way, for sure, and there have been. And but we haven't given up, not given up. And what takes so long, and we've been at this for close to 8 years, is that you got to bring all of these elements together. And at the end, again, I emphasize, have it at very low cost of our target of $50. And this has been what we have been working on. The work has resulted in a portfolio of 58 patents for the company and a bunch of technologies that have been created and everything else. But of course, we're not there yet. To show a couple of examples, the first 4 cancers that we have focused on are lung, breast, prostate, and colon. 3 of those, as I said, early detection is a major lifesaver, and that's why we have selected those. But those 4 cancers actually constitute more than 50% of all cancers in the world, nearly 50%. And that by itself is a major thing if you can accomplish it just with these 4 cancers. But the goal is beyond this. We do have not not blood data, but tissue data from 17 other cancers, including all the major ones, including pancreatic cancer, which is a big killer and difficult to deal with. And I am just jumping around in its slides to show a few examples of, no, not that. So if you can see this chart, this shows some data on serum samples, on blood data from those 4 different cancers with those number of samples: 60 lung cancers, breast 181, prostate 233, colon 145, and 218 healthy subjects. And this shows the collective data in here shows a false negative of 4.4%, in other words, sensitivity of 95.6%, and a false positive of 7.8%, or a specificity of 92.2%. These numbers for those cancers are actually great numbers. If we can actually achieve this, this has been in the form of a lab test, not in the cartridge test. If we can really achieve this in the cartridge, it would be, I think, phenomenal. We're trying to make it even better. But something else that's interesting about this data is that instead of showing it as a collective graph like previous slide, if you do it for the individual cancers, each one of those plots, and the individual stages 1, 2, 3, and 4, that at every stage of every one of those individual cancers, it has the same performance. What that tells you is the potential for early detection, which is, again, one of the goals here, right? And that's kind of a little bit of a more exciting part about this than actually being able to say we detected cancer. So combination of all of these together, and if I can show perhaps one other slide, is maybe this slide. So I call this 7.5 years and 320 slides compressed into one, because this puts together all of the elements that make this possible that we have to go. Now, if I were to give a talk on this, it would be at least 10 hours, all right? And there's no way you can cover all of that. But I'm just pointing out to all of the main elements, starting with an antibody-antigen pair that we track. There are pieces of cells that separate from your cells, enter the bloodstream, called exosomes, extravesical. And these exosomes are about maybe between 50 to 150 nanometers in size. They carry a lot of information, a lot of biomarkers, all kinds of genetic data, extremely useful, and you get about 1 billion of them per milliliter of blood. Our work focuses on that. There's a new exosome assay that we have developed. There is an antibody that is highly specific to a certain target that's common in all cancers. That we track this antibody purely out of luck. We have the exclusive license to it in perpetuity, and this I would not be able to do today, but 8 years ago it just happened because I was so lucky. And this antibody, we actually produce it, we purify it. The purification method is very difficult and a trade secret of the company. And that's one of the elements that makes this possible. On the biology side and biochemistry side, there is a special exosome assay that has been developed for doing this detection. We have a lateral flow assay. Some of you, if you're not familiar with the term, I'm sure you've used it because it's the same technology that a COVID test or a pregnancy test or a fertility test uses, with a few drops of a solution in the COVID case, mostly the nose swab, that detects the virus. And this technology, lateral flow assay technology, it is cheap and it's very rapid, right? So a question should be asked: why can't we use this test for many other things? And because it has a couple of very fundamental limitations, and those limitations are things that we have essentially thrown technology at it to solve, and have created a new platform for doing these tests with lateral flow assay, which by itself is, as I said, a platform test technology independent of the cancer work. This could be used for many other targets and molecules, and we are in the process of trying to commercialize this so it can be licensed by others. There's no way in the world that we can, being a small company, try to do all of that, and we're not that naive to think that we can do that. But we can make the technology available for licensing. There is a certain microfluidics technology that we have also created as in parallel path. Those of you who might be familiar with microfluidics, if you have enough money and you don't care about how expensive your device gets, you can build any microfluidics you want, no problem. But if you're going to build microfluidics that you're going to use as a home test cartridge and be low cost and manufacturable in volume, everything else, totally different story. And integrate sensors with it. And that's an area that we have also done a number of things. We called it MicroLab AZ because it handles everything from A to Z on a little cartridge. And that is a parallel track to that lateral flow assay technology that I mentioned. And that uses also a biotransistor technology. That's a joint development with another company. And in order to increase the sensitivity of the detection with the sensor and all the sensing electronics and everything else integrated in a cartridge in a disposable fashion. And then we have certain distribution tracking for the whole test. And I mentioned a patent portfolio that makes this possible. So I'll talk for another minute, then I want questions. So first question is, so where are you with this, all of this great stuff? So we are at a point that we conduct tests in our lab as a lab test. And as of about a month ago, we started to do it in live subjects, not samples that you buy from sample banks, not frozen serum, fresh blood, fresh subjects. And that is happening actually right now. That is really to optimize the assay and everything else. Within 2 years, hope to be able to offer this as a lab test some of you might be familiar with the test that's out there on the market called Gallery from a company called Grail. They have been on the market for a while, and they offer this test that's not FDA approved, but it's what's known as CLIA approved, costs $950, and you can do this lab test that for early screening. It has a sensitivity of 50%, but a very good specificity of 99%, and we are an advocate of them because our work is totally complementary to what they're doing. Ours can reinforce their results, theirs can reinforce ours, and that test has been available. Insurance doesn't pay for it yet, but you can take that test yourself if you want to go arrange for it. We hope to do have a lab test in 2 years as a product, and at that point have the cartridge ready for launching the big FDA trial. We are quite aware of the fact that this is a very challenging FDA trial. It's going to be very costly. We are planning for it. We work with companies that develop regulatory strategy. We're not all alone in this. And I'm quite aware of the amount of money it's going to take to do that. So all of this stuff, the success of this depends on how also successful we are raising the money, which is an important issue you have to keep in mind for any startup company, regardless of how good your stuff can be. Even the buzzword AI that used to get you all kinds of money these days has a problem. I've seen a lot of companies literally use it as a buzzword. Anything that has an if statement in it is AI, because it makes a decision. All right? That's kind of where we are. And of course, it still has its risks, no doubt. But that's what makes it challenging and fun. And in order not to get totally consumed by this, on the side, I designed 28 gigahertz phased array antennas and 6 gigahertz range antennas and stuff like that. And that's kind of like a little hobby, but at least if this doesn't work, it will work. This is my passion for life. But I can have a backup and also try not to stay away from the circuit stuff. That is one of the elements of happiness. Okay, so now questions. I need you to ask difficult questions. Yes, sir.
So you seem to have found the 2 Pi Sigma company in September 2016. In September 2016, right?
After 16, yeah.
So I think at that time was quite close to the fall of Theranos. So what's your opinion or like lesson learned from the Theranos?
Right. So when Theranos started with what they said was years back, I was actually first time we heard about what they were going to do. I was sitting in the office of a biologist that used to be a close friend in Maryland, and we both said, that ain't going to happen, all right? And okay, so she was trying, or Theranos was trying, maybe all started with good intentions, but then got into a track that they couldn't get themselves out of trying to do everything with a single drop of blood, not to do one specific thing, all right? And that someday could become possible. We might be getting there at some day in the future. But back then it was totally, we knew that it wasn't gonna work at all. And that was the main issue with it, where the claim was from this one single drop, I can do all of these tests, all right? So the fact that they failed, of course, and at some point as a private company, they got to a $10 billion ridiculous valuation. And she managed to get some important people on the board of directors that made it more and more believable. And they got themselves into this vicious loop of having to lie and lie and lie and never get out of it. But no matter what their what the cause was, no matter what the facts are, whatever it is, it has hurt people like our company. And so this one of the unfortunate things is she always wore black and I always wear black. But I used to wear black all the time before she did. Right? So I did have on one of my early slides is that even though I wear all black, but it's no correlation there. So you should expect people to be skeptic about it, right? And you can't fault them for that reason. And to answer your question, that was actually one of the reasons that we didn't start with. As I'm saying, to this date, there's no website to tell you what we're doing, and never advertise nothing. So that's the way. But you have to deal with that fact until you really have your final product in hand. Yeah.
Yeah, Mehdi, thank you very much. It's very interesting talk. Thank you for your passion about working on this very important
Thank you so much.
problem. Whatever progress you make is a progress. It's a very important step.
Love to hear that.
So thank you for that. But my question is, so as you are working, you have different types of tests that you are developing. Let's say exosome assay or antigen pairs, and are any of these things new, or these were known and you're not trying to apply it to your small technology?
No, there is a basis. There is 20-some years of work prior to it, which also had resulted to that antibody that I told you, that we ended up licensing from the pharma company that was working on that. And there is quite a bit of work. Like, for example, there was a table of tissue samples from 1,500 patients with 17 different cancers that shows the prevalence of what we are trying to detect in these cancer patients. That's tissue samples, it's not blood yet. Blood is only those 4 so far. We don't have data on the rest. And no, there's no way we could have done all of this just from scratch, everything from scratch, obviously. But yes, there's prior effort on it. And what we are trying to do is to bring it all together. And in the process of that, we have created a new technology, we have created a new assay, and all of that just from the attempt.
So if you were to separate the $50 cartridge and then a clinical trial, so I could go to a clinic, take a blood sample, go through these tests that you have, and be able to detect early or not.
Yes. Yeah. So, but in order for me to assure, we do have volunteers who come in. Okay, the very first live patient, the fresh blood that we did, came out positive. Right? There's a form, there's a consent that they sign that this you cannot be used for making medical decisions. This is for research purposes only. That person didn't make medical decisions based on this test, right? But 10 days later was diagnosed with early lung cancer, was operated on 2 days after that, right? And it was so contained that he didn't even need chemotherapy, right? Now, that's one data. I would never use that one piece of data as any scientific basis at all. It's just an anecdotal piece of data. And but in that case, the person did have nodules in their lung, one nodule in the lung that wasn't characterized as cancer. So anyways, 2 weeks later, he went and did biopsy, and then they verified it. But before that was done, so the test came out positive. That we do in the lab now, but again, as I said, you cannot make any medical decisions on it. And that's just for collecting data and collecting assay. But yes, it will be possible to do that soon, or in a couple of years, to go to the lab.
Can I follow up on that? So,
Sure.
Why did the patient have a biopsy? Did they go have another CT scan?
No, beyond, just because of the nodule in the lung,
But
that
how
the
did they
scan,
know?
CAT scan. That was a CAT scan, right?
Okay.
So,
A CAT scan that took place after
no,
your...
prior.
prior.
He had a nodule,
Oh, I see.
so he came to us to kind of test our scheme and everything and all that
I
before the biopsy.
So you
Because
went-
if he came to us after the biopsy, we are not biased with the
yes,
results, right?
Okay, right.
So this is prior to it being verified as a cancer.
Oh, so it was planned to have a biopsy all along?
He was planning to have biopsy, but purposely did this
I see.
test
Okay,
prior
so
to
good-
biopsy.
good subject.
Yeah, that was a good.
Because a biopsy has
Exactly.
its own risk.
Absolutely, yeah. So this was a good accidental subject. Not that we are happy that he had cancer, but
Right.
so yes.
Yeah, so this is, my background of strong EE, little biology, but I'm a little surprised. So is it actually the case that a lot of cancers have these molecular markers that are bloodborne that you can detect? That's surprising to me.
Okay, it should be surprising to you. There are not too many screening tests available, right? And there is a lot of work on genetic markers. Okay, for example, the work that company Grail is doing is based on what they call a methylation. They do sequence your gene, go through all the stuff that the Illumina's machines do. They find this methylation that says yes, indication of cancer, but that pattern of methylation also is an indication of what type of cancer it could be, and that's their approach, which is genetic approach. In our case, it doesn't identify, it just says cancer or not, right? And then once it is positive, you have to go after finding the tissue of origin. And the whole idea is that this is a very low cost test. Majority of people will not, 0.35% of population has cancer every year, something like that, right? So if this is a test that everyone is doing, is going to be only a small number of people that become positive. So when you get to that stage, by doing this cheaply, that's the point to now focus on what it is, do PET scan, do that, and identify both the source or also take these other tests. But the whole thing is to catch it first at very low cost. And also periodically, because once you establish a trend, if you do this test for 3 years, every month you got these 36 data points and they're all negative. Once all of a sudden you have 2 positives, you've caught something there because you establish a trend. PSA does that, for example, for prostate cancer, where the trend is more important than the actual absolute value that you test for. Yeah.
Can I follow up on that?
Absolutely.
Because I think, so you're targeting 4 cancers. Are all 4 of those chosen in part because they do exude some chemical that you have a chance of detecting?
No. The 4 are because they are the 4 major ones, right? And the 3 of those are stage 1 is life-saving. It's not extend your life for 2 years, okay? And lung cancer is stage 1, 60% survival, 5-year survival, right? But breast cancer, 99%, 100%. So it is that first of all, they are the first 4 major ones. They are more than 50% of all cancers, and 3 of them have the potential of totally saving your life, and that's why,
Okay, but to detect it, there has to be something in the bloodstream through your system.
Right.
The question is, I think of a lung nodule as a contained little thing. I'm probably wrong, but that's how I think of it. So maybe to give in a drop of blood, how many?
1 billion exosomes.
Oh wow,
Yeah. It's not like circulating tumor cells are 100 per milliliter maybe,
Okay.
but exosomes are 1 billion per milliliter.
Okay, maybe I'll ask, what's an exosome?
These extravesical, these are pieces of cell that literally separate from your cells, enter the bloodstream. They carry biomarkers, they carry genetic information, and so that's that. Circulating tumor cell is a different story. If you catch a circulating tumor cell, you can definitely characterize and learn a lot about it. There's a lot, as a matter of fact, there's some, you work at U of M on this, I think, on the circulating tumor cells, if I'm not mistaken.
So you're not trying to catch those cells, you're trying to
No. Dave?
Yeah, I was gonna ask if there's other body fluids that come close to blood in terms of a carrier of
Exosomes.
information that you could use.
Urine.
Swabbing.
Yeah, urine, probably saliva to a lesser extent, but urine is another potential candidate that we are actually going to also start doing work on and studies on. But given the budget we have and whatnot, of course, you can't do everything at the same time. But urine is also known to be a good source of exosomes, whether it has the exosomes that we look for is something that has to be established. We have to do, but urine is an example, yes. Back.
So the patient that you had, you said they were planning to get over. So, did you ever get to test them after they got cured?
Yes.
And it was negative?
And no, it shouldn't be because that lingers for a while and scheduled to do it in touch. But it was, although we do not characterize this test as something that quantitatively stages cancer, right? If you remember that actually plot, I showed you that at different stages it was having the same performance, but his results were slightly actually lower, although would still be considered a positive, but that is also expected for 2 months. And so he's scheduled to come in a month or so we can do it again.
So you expect that one to be
That one we expect to be negative. Yes.
I mean, all cells in the body, you said, release exosomes. So how are you able to get that specificity for a small human cell that's very tiny compared to all the other cells?
Right. So because we look for a certain thing on the exosome. So there are 2 conditions that have to happen. And that's actually one way to increase our signal to noise ratio, because what we look for can exist as background in your blood. But if it is on exosome, we track it. So that's how it gets specific to cancer, but it's very difficult to detect. Hence all the calisthenics about trying to detect this very sensitive circuits. So yes, that's what was.
It's not independent. It's not dependent on which person it is. Everyone with the same kind of cancer has the same kind of biomarker. That
It is a general amongst. I told you the 17 different cancers tissues. They all look for the same thing. And so does this blood test that we have, same thing. That's why then it's a screening, not a diagnosis. You screen for yes or no, and then once it is positive, now you can't do that kind of screening for $1,000 test. That will not make sense in that way, in my opinion, that only says yes or no, right? For a test that does say which cancer, yes, maybe you can spend that kind of money. But to just say yes or no on every month, it has got to be very low cost. And then when it's positive, now you go after where it is and chase it with other techniques, which will be more costly. Yes.
Let me ask you another question. In your screening process, do you have a knob that can adjust between false detection
Yes.
versus false positive?
Yes. So those can be traded off against each other. It's simply a threshold number. It's just a number. And you can go to, like, for example, the data that I showed you, because I've done this exactly. I mean, the table is actually there if I find it. So you can go to only 1% false positive or 99% specificity. Their sensitivity drops to 70%, right? On the other scale, you can go to very low sensitivity, but then the specificity gets worse, you have more false positive. Now, as far as you and I are concerned, we want a high sensitivity. You want low false negatives, because if you're negative, you want to you want to feel good that, hey, it's negative, right? The false positive is something, the low false positive is something that the insurance company wants. Because whether you're true positive or false positive, you still have to verify it. You have a mix of false and true. To separate those, you still have to go through this for So it doesn't matter that 0.35% of the population are positive. Those are your, say, true positives. But if you have a 10% false positive rate, your rate is really 10.35%, and out of that you have to go through the cost of identifying which ones are false positive. Which, for example, PSA test has a very bad rap. I think a lot of people believe PSA is bad. Actually, it ain't that bad. Why? PSA has a sensitivity of 93%, and it's 7% false negative, which is not bad, right? But has an awful 80% false positive, and a specificity of only 20%. So that 80% false positive, then after examination, this and that, you go for a biopsy, which is not a straightforward procedure, okay? And it is that biopsy that a lot of times shows that it was negative, but it does find then positive ones as well. And that's the cost that the insurance company has to go through. And that is why not every single false positive from a PSA test really gets automatic biopsy. You now have to have more reasons for doing that biopsy. There'll be more examinations. Your doctor has to really be sure that you need a biopsy, and then you'd go do a biopsy. And we actually have a scheme to combine tests and increase that 20% specificity to 84%. But that is where the thing comes from. Yeah, Dave.
Yeah, to follow up on that, I was wondering whether there's a single threshold, or are there a number of thresholds that contribute to the final one, and you could imagine that the other thresholds have to do with other parameters about the patient and so on.
Okay, so you're not reading into what we are exactly doing, in that there are other parameters involved, and there's a little bit of what I call mini AI involved in there as well, that you got to optimize across all of these a few parameters. That, right? But at the end, though, you cannot then, once your test is established, you now cannot change that. Like from subject to subject, it can't be variable, of course. So you have to make it still results in a trading off false negative versus false positive. And once you arrive at what you want it to be, then that is fixed. And that's what you propose to FDA to try, not to change it every single time. And that is, so if you do a test, for example, 3 times in a row and try to average and whatnot, that doesn't catch any biology related thing. That could help you with your system errors, right? Anything that is systematic in your noise, in your detection, in your circuit, in your optical reading, in your procedure, the person who is actually doing the test, it can catch those. But if a person doesn't show this marker or shows this marker, no matter how many times you test, that accuracy is not going to improve by repeating this test in that way. You just get rid of other random things that might have happened, or some noise in reading. And so the test repeats in that sense, are not going to help a lot, but they do help with reducing noise. So, yeah.
I have 2 questions. One is technical, the other is non-technical.
Okay.
Technical
We charge you for that one. So, yeah.
You said it's a $50 cartridge. But it works by itself, or you need a reader, instrument, and machine, which can
No, you don't need the machine. You need a box, which costs maybe $40, and it can be given out for free. Right? Because you're doing this every month. Don't forget. So that's not your box. Is not the revenue maker. Is the ink. The ink and the printer is that thing that gets you, right? So that's the approach. Yes, that's the famous razor blade model. So yes, you do have to have that quote-unquote control device reader. The little box that you saw on the first slide, that is now in its 4th generation of design. So, you need just like a glucose meter, right? So, similar. Second.
Where was 2 Phi Sigma come from?
There's Jeff's question. So when I started the company, I wanted to call it Six Sigma, because what you're trying to do is like way out there, it's a big spread, and Six Sigma was taken. And I was having lunch with a Broadcom colleague, and he said, well, 2 pi is more than 6, so to call it 2 Phi Sigma. So, and it's stuck, yes.
Are there any other diseases that you're looking at that you could use this technology to diagnose?
No, but definitely the platform has the potential of doing that, and that is what we're trying to commercialize as a platform.
Any
Yes.
potential diseases that you have?
There is some heart biomarkers that could be a potential, and there could be a ton of other things that I'm not even qualified to answer, but I am certain. All of the other tests that you do, a lot of them are this kind of ELISA type test, and can be converted to this. But each one needs a study, needs a FDA trial, needs money. So who said money doesn't bring happiness? Yes.
I have a less technical question. Like, you've seen pharmaceutical companies that they have products and then they end up jacking the prices. Do you have a plan for free investors or something, tell you to raise the price of the product, like to increase profits?
We don't have any plans like that, obviously. And what is lucky about this thing is that the insurance companies are going to welcome this, because of the fact that if you detect early, it reduces the cost of the treatment to begin with. And also, if the test is very low cost, so our goal is we're not even counting on insurance covering this test, okay? There will always be people who would pay the $50. As a matter of fact, we have a portal that will take care of this. It's kind of like a service. You pay $50 a month, you receive a cartridge. You do the test, your test is tracked, all of that. So those are our first set of customers, right? We're not here to all of a sudden say we're addressing 100 million customers. Those will be the first set of customers who are willing to do that until this thing shows its value. And that's a point where insurance company, Medicare pays for the Cologuard test now, right? And which is a cancer screening test for cancer of colon, of course. So that's our target or our approach to market this. Okay? Khalil.
Thank you again for the talk. So of all the technologies that you developed, you had that list of all the different technologies. Which one would you say was the most important? The one that really kind of did allow you to kind of make this whole system work? Is there one thing that was really the most enabling, in a sense, the most disruptive?
On the technology part is the lateral flow assay. Okay. Of course, that relies on that antibody to have that, but it's the lateral flow assay, which I said it turned into a platform technology that could be an enabler for many other tests. So that, I would say, was the most significant one as far as the goal of making it possible being low cost. That would I call the most significant one. Yes.
Nice talk. Thank you very much.
I haven't talked much about this.
When you're saying early detection, early diagnosis, so how much early, like with symptoms, or even persons without symptoms can also?
Okay, so the only thing we can prove that is feasible to prove is a stage 1, obviously. Why? Because to do any trial, you have to have positive subjects, and you have to have people who are diagnosed at stage 1, and you detected all of that. But if you also want to do a long-term study, which would be extremely costly and extremely long, would be to follow a crowd of people who don't currently have cancer, do this test regularly and see if you catch all the stage ones and whether prior to symptoms you catch all of that. So while the potential is there, if somebody asked me to prove it and show the data, it's not there because that's going to be very costly and long term. So all of these tests that have that have to go through that step. Of course, if you want to show that you caught it pre-symptoms, right? So.
So we've talked here a lot about the biology. Could you just say a few words about the actual electrical and computer engineering part for this audience? Just to have, even if it's in standard, you know, I have no idea if there's a laser or a diode or you know,
Okay,
what's
all
inside
right.
the box, just as a run, you know, for this audience.
Okay, no worries. So on the part about the electronics and the electrical engineering part of it, that lateral flow assay technology, I'm wondering whether, well, okay.
Sure.
I want to find this if we got time.
Yeah, we got a few minutes.
First of all, who knows how LFA, lateral flow assay, works? The COVID test that you have, how many of you know how it actually works? Okay, she knows. All right, don't say it. Okay, so let me do because, okay, so that's important. This is a COVID cartridge, okay? So you've all done it, and next time, if you have a cartridge, split it open. It's very easy, a little sharp knife, it will split it open. You see that sliver in here, this particular one is 65 millimeter by 3.5 millimeters, right? This innocent looking thing, it's very cheap as well, is 4 different pieces of material labeled as such: sample pad, conjugate pad, membrane, and absorption pad, okay? And on it, there is chemistry, okay? That does this detection, and at the end, you see the lines, right? So how does that do this detection? There is let me use my finger as pointer. There is a drop of whatever solution you have that contains your marker, whatever, that sits on this sample pad, right? Whatever you put on this with capillary flow, just like put a little water on a napkin, it's going to start flowing. It starts flowing that way, and hence the name lateral flow assay. So this sample comes over to this other pad, is called the conjugate pad, right? The purple material in the last slide is the chemistry that's on this conjugate pad. This conjugate pad, on it, it has this antibody that is specific to your target molecule. In the case of COVID, there is, on the virus, there is a protein called S protein that's very prevalent, and there's plenty of it, luckily. A lot of people don't know how lucky we got. That protein is in abundance that made this possible. Within a month, people could generate this cartridge. This is a very lucky event that people just do not realize. Otherwise, you would not have a COVID cartridge for general mass screening. This antibody is tagged with a certain tag, in this case, a 40 nanometer gold nanoparticle, right? One of the properties of that gold nanoparticle is that you can easily do this process called conjugating, attaching these to the antibody. If the solution that you put in here contains your target, that target is going to come stick to this antibody, right? But not all the antibody molecules are going to be used, right? There will be some that don't stick to anything. So you will have a combination of this antibody that is on your target, with your target molecule, and some that are just free like that. The reason they were tagged is because to be able to track them, to measure things, to see them. This combination of this solution, complex solution, flows over on this strip, which is called a membrane. It's a nitrocellulose material. It's a special material. It's not, people call it paper test. It's really not paper, it's membrane. And this thing flows like that, laterally, the whole thing. So on this area, which is called the test line, which all of you have seen, there is another antibody for this target, different than this antibody. You have two different antibodies that are sticking to that molecule. Assume that you have that. So there's another body that, quote unquote, is immobilized, means that it is sticking to that surface. Whatever sticks to it, it stays there. It doesn't go. So the molecules that have your target molecule and the tag stick to this and stay there, right? The molecules that are just the antibody obviously don't stick here and they continue flowing, right? Now, before the test, the ones that continue flowing anyways, regardless of your target and everything, come to this line. It's called control line. This control line has another chemistry and another antibody that captures any antibody. It doesn't have to be any specific. That will capture this antibody that happens to have the tag on it as well. So they concentrate here, those gold nanoparticles, when they concentrate, they turn red. The more you have, the more of a contrast you have. COVID tests, they tell you even if you see a faint line, you still have COVID. If you see a very dark line, you really have COVID, but they don't, but they don't classify it like that. They're not trying to measure the stage of you and whatnot. But what that tells you is that's why it's called the control line. That tells you your test did finish properly. If you don't get that control line, something was wrong, because that must show up. That's why they tell you, if you don't get a control line, test is not valid. Once you get that control line, and then the rest flows, and this is called absorption pad that wicks it through. Once that lights up, and then you're looking at this test line, and if, again, you had the target molecule, in this case, the S protein of the virus, this starts changing color, and you see all of those colors, all right? You see how much easier it is for me to explain this to a crowd that has used the test? Because you all have used it. You just, you understand what I'm talking about totally. And, okay, so this is the whole process, as simple as that, right? This entire thing costs, I was involved in costing one of these for manufacturing, maybe $2.75 for the chemistry, the plastic, the strip, everything. It's very, very cheap for the cost of material. So this, right? So the question should be, why can't we use this for all of our tests, right? Well, it has two major limitations that reduce its sensitivity. One is, when you get here, you're at the mercy of how long this material holds that solution before it passes on to that. You have no control over that. It's a piece of material. In a few seconds, this thing mixes and goes. But if your particular molecule needs more time to bind, in our case, it needs 5 minutes at least, then nothing will bind and you will detect nothing, right? So that's one thing, incubation time. When this thing flows over, if this test line, which is 1 millimeter wide, all the time that this has to capture it is when it's crossing that test line. And if it goes fast, again, you don't have incubation time. So because this flow rate, right, that there's no way to control it. It just flows. And as a matter of fact, when they sell these membrane materials, when the manufacturer sells to the people who make the test, there are different grades that they call them high sensitivity, low sensitivity. And guess what? The only difference is the flow rate. High sensitivity has a slow flow, low sensitivity has a faster flow. But the range is not that much, right? So, okay, all of these limitations. Now imagine the part that I'm not going to completely explain, that somehow magnetically you can control this flow digitally. I'll say how long I want it to stay here, and I say how slow this needs to be. Minute or half an hour. and you can program that. So that's a little bit of magic there. That comes into the electronic part and everything else. You have to come be hired at our company so we can tell you. But that's one element now. Then there's other stuff about microfluidics is also the same way.